Biospectra-by-sequencing, it's the name of the project that I'm going to call BBS, otherwise I'm going to run out of time pretty soon. It is supposed to represent a workflow here. Input, what's happening in the middle, and output at the end. The Biospectra-by-sequencing project comes from something was called genotyping by sequencing before BBS. That was a new method for sequencing organism developed at Cornell University in the New York State. Basically what it gives is a very cheap and efficient way of sequencing only part of a genome. The interesting part is that if you use the same restriction enzyme, you always get the same site from the same species, and then you can compare and do population genetic with those data. It was firstly used on maize because the population genetics on maize has been well studied. They've got lots of pedigree data and lots of things to work with. So as it shows on the diagram, you get always the same sites. You can compare for variations or for insertion deletion indels if you've lost a site. This technique, while they were developing the wet lab part, also developed the analysis pipeline at Cornell. There's basically two pipelines now available from them. One if you've got the reference genome of your species, or one if you don't. Of course, if you've got the reference genome, it's much easier to get reliable and more results than if you don't, but nonetheless, no reference genome doesn't prevent you from using this technique. So that was the input. Now what do we want to do with this? Because if I'm just stopping here, you'll think, well, there's a software, there's a technique. What are we going to do with this? That's not a problem, but that everything that happens in the genomic area is that the technique is not used only for genotyping anymore. It's been widely used for other techniques such as finding new genetic markers. Whatever you can think of, people are going to use this cheap and efficient way. Even if it's just a sub-part of the genome, it's usually enough for people to have lots of data anyway and to do something with it. So that started in New Zealand with Robert Shire coming from Cornell to New Zealand and trying to create a community of people who would work on all this new application and try to work together to make available new analysis pipeline, starting from Tassel, but expanding it. So we could analyze all this other data that were coming from derived application from the original BBS thought. Because we've always talked about reproducible research, when I'm not going to go into it in too much details, but that's something that's really important to us, is that we can redo the same thing that we've done before, because if I go back to something I've done 6 months ago, I won't be able to rerun it the same way. If I don't have something robust to rely on, proper workflow, framework, whatever the word is for this. And also because most of the scientists we work with have published their data maybe a year after they've actually produced it. And same thing, we definitely need something that we can go back to and help them actually saying what they've been doing. So what do we get at the end? We get results. We get what we call metadata that we want to keep. That's very important to us. And what we're trying to do is a community toolbox. There are two aims to this project. There's for bioinformaticians who analyze this data to have something they can rely on and have those tools and make those reproducible research workflows. But there's also the aim at making it available for the community. And I think that's another topic that has been talked about here, about how do we include scientists into this bioinformatics and teach them how to use them. But that's something that we think at least some scientists would be keen and could do, is that if we provide them something that's all packaged and they're ready to use, they could actually analyze their own data. That's what Biospectra-by-sequencing is. It's a project that aims at getting a community together. So bioinformaticians get all the knowledge they need and the help from the community to make all those tools available for this data that we have now. And it is an effort in the community also to make available those tools to anyone who would be interested in analyzing their own data with this. And that's all the people that are involved at the moment, and anyone who is interested is most welcome. Thank you.